Hello everybody and welcome back to microbiology. Today we'll be discussing microbial genetics. To do so, we'll have to first understand what genetic material is. And genetic material is very important because it defines the structure and function of microbes. All living organisms store their genetic material as DNA. However, viruses store their genetic material as DNA or RNA. So let's do a little review of our basic biology and discuss how the structure of DNA looks like, how it's packaged in prokaryotic and eukaryotic cells, and what are the essential functions of DNA. DNA is made up of building blocks called nucleotides. Nucleotides are made up of three different molecules that are all interconnected. One is a phosphate group, which has a phosphorus atom in the middle, and it is surrounded with oxygens. This is linked to a sugar molecule. The sugar molecule in DNA is deoxyribose. Ribose is a type of sugar just like glucose or fructose. And it is the deoxy form. That means that one of the oxygen groups has been removed. So instead of having an OH on this second group here, it just has an H group, and we'll discuss that in more detail later. Finally, all of these have a nitrogenous base attached to them, sometimes just referred to as a base. There are 4 bases in DNA that make up the code. The first one listed here is adenine, abbreviated as A. The next is guanine, abbreviated as G, cytosine as C, and thymine as T. These building blocks are linked together through what is known as a phosphodiester bond. So remember that we had a phosphate that is attached to the sugar, and these phosphates act as a bridge connecting one nucleotide base to the next. So if we examine this phosphate group in between these two ribose sugars, we'll notice that on either side of the phosphate there is an oxygen that links to the other sugar. And this is called a diester bond because this OO structure here is an ester, and there's 2 esters on either side of the phosphate. So this PO2 is one of the esters, and on the other side is another PO2 for the second ester. What happens is cells can take one nucleotide, and through a reaction called a condensation reaction, it can create take two molecules and combine them together. It's referred to as a condensation reaction because just like condensation, when you think of rain, the product of which includes water. So it removes one hydrogen from one of the riboses and an OH group from one of the phosphates, and we're just left with that single oxygen that remains in the molecule to create this diester bond, and a H2O recombine from these 2 original molecules, and so we get a little bit of water, 1 water per bond that is formed. So if we wanted to build a DNA molecule from scratch, here's how it would work, starting with a single nucleotide. Here we have a adenine nucleotide, and attached to the 3rd carbon of its membered ring, 1st, 2nd, 3rd carbon, we have this phosphate that attaches through a condensation reaction. And so we've attached a cytosine here. And this can be done again and again, a guanine, a thymine, and a cytosine again. And as you can see, no matter what nucleotide base we use, we always have the same sort of reaction and bridge this phosphodiester bond that links the 2 together. As this is accomplished, a new structure emerges from this DNA that is formed. This structure is referred to as the backbone of the DNA. So on each strand, you can imagine that there is this phosphate group and a ribose group that are attached to each other, alternating between each, and this makes up the backbone. And another piece of DNA can bind to the first piece of DNA, and this creates what we call the double helix. Something that's interesting about these nitrogenous bases in the middle is they have these extra hydrogen and OH groups, hydroxyl groups, that are available to interact with another nucleotide base. And their structure and spacing is thus that an adenine can hydrogen bond, is what this is called, when an oxygen and a hydrogen interact with each other. An adenine can hydrogen bond to a thymine, and it does so with exactly 2 hydrogen bonds. And again, a cytosine can interact with a guanine, and it does so with its 3 hydrogen bonds. So no matter what, we can only have an AT or a TA pair up with each other, or a CG or a GC pairing up. And remember, this is called a hydrogen bond that occurs between these 2 strands. These hydrogen bonds are not as strong as these other bonds we've seen in the rest of the molecule. The other bonds here are called covalent bonds, and they're a lot more sturdy than the hydrogen bonds. And you'll see that that is very important when it comes to using DNA in order to accomplish the production of genes and replication. And so together, the 2 strands are called complementary strands, and they always end in opposite components of the backbone. One ends in a sugar and the other ends in a phosphate. So you see this first strand at the very bottom component is a sugar, the ribose, and in the other strand, which has to go in the opposite direction, will end in a phosphate. This has to be done this way in order for these hydrogen bonds to match up. Now, in reality, the DNA does not sit flat like this. It has a 3-dimensional structure, and it looks more like this, and we're all familiar with this drawing, this cartoon depiction. And this is what we call the double helix. So it's double because there are 2 strands of DNA, and we can see that the G's bind to the C's and A's bind to the T's. And we have the backbone is drawn here, and this backbone, you shall remember, is the phosphate and ribose sugar that line the backbone. And we have something called the minor groove, which is the distance between the backbones here, and the major groove, which is the distance between one backbone to the lower backbone here. And so this creates what sort of looks like a spiral staircase. Now let's discuss further about the numbering system of this sugar and how we use this to understand DNA. So if any sugar that you take, it has a series of carbons within its ring, and here we have 1 extra carbon that lies outside of the ring. And scientists have to have a way to identify which carbon they're talking about. So they decided to name these carbons. If you go clockwise from the oxygen, they start with the first one. So the first carbon is always going to be the one closest to the O in a clockwise circle. And so this first carbon is labeled 1 prime. The second carbon is 2 prime. The third carbon is 3 prime. The fourth is 4 prime. The fifth is 5 prime. And if you will recall from your basic chemistry courses, any of these points here that you see, or when 2 lines come together, that represents a carbon that exists there. We just forego writing carbon so that we don't overcomplicate the drawing. So now we can start to talk about the directionality of DNA. As you recall, we have a phosphate group that's bound here, and this ribose can link to some other nitrogenous base or some other nucleotide below it. So this oxygen is available and drawn here with the dotted line, but not included, to bind to another phosphate group down here. So this own nucleotide has its phosphate group bound to its 5' carbon. And it has the 3' group available to bind to some other nucleotide's phosphate. So when we look at this image, we should know that every 5' group should have a phosphate bound to it in a nucleotide. And if we want to bind another base to this, we can bind it to the hydroxyl group here of the sugar and form another phosphate down here and form a phosphodiester bond. Or we can bind this base to some other sugar above it. So you should be able to identify which carbon is the first, second, third, fourth, and 5' and what should be attached to it. For example, the first prime should always have the nitrogenous base bound to it. The 5' will have a phosphate group bound to it. If this particular base was at the end of a DNA strand, then the 3' would be an OH group. If it was in the middle of a DNA strand, then this 3' end would be bound to a phosphodiester bond. So, checkpoint 1. Does the 5' end or 3' end of a nucleotide have a phosphate group? And this is having to deal with a single nucleotide. So scientists have learned a lot about DNA based upon its structure. Before DNA was discovered, a man named Gregor Mendel had associated certain phenotypes with what he referred to as genes, and he believed that genes were discrete units that were heritable. However, scientists did not know what the molecular mechanism was, what the source was for these genes. And the most common explanation was that these were from proteins, because there were many, many proteins that were found in cells, and so it was thought that that could explain this sort of heritable trait. Well, along came a few scientists, and you might recognize some of these names. We have James Watson to the far left, and we have Francis Crick standing next to him, and Rosalind Franklin and Maurice Wilkins. Now, James Watson and Francis Crick were biochemists, and Rosalind Franklin and Maurice Wilkins were really good at X-ray crystallography. In particular, Rosalind Franklin had a lot of contributions to the understanding of the structure of DNA. So, as you can imagine, cells are very small and difficult to observe underneath a light microscope. And so to observe DNA, which is much smaller than a cell, these scientists had to use something called X-ray diffraction. And what you're looking at here is an X-ray diffraction of DNA. And Rosalind Franklin kind of was a artist at doing these sorts of images. It took a little bit of talent in order to get these really sharp images. And from these images, Francis Crick and James Watson were able to discern the structure of DNA. And here, I believe it's Francis Crick. I believe this is Francis Crick standing next to a model of DNA. So from that image like this, they were able to decipher that the DNA had a phosphodiester backbone, that it had nitrogenous bases in the middle. They realized it had a double helix that had a minor groove and a major groove, and what the nitrogenous bases were. So You might wonder, all right, we have the structure. How do we get from the structure and from that derive the concept that DNA is where our genetic information is stored? And I pulled up the original paper that was published in 1974, and it's entitled Molecular Structure of Nucleic Acids, a Structure for Deoxyribose Nucleic Acid. And here is their own original figure, their cartoon depiction of how this DNA molecule should have looked like. And as you can see, they even have this anti-parallel complementary nature drawn in the backbones, where they knew that one strand had to go from 5' to 3', and the other strand had to go in the opposite direction, 3' to 5', in order for those nitrogenous bases to hydrogen bond to one another. And they have a very interesting quote here in their discussion section. In the discussion section, often you will have scientists who provide the implications of their work and where further research should be done. And in this discussion section, they say, it reads, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Full details of the structure, including the conditions assumed in building it, together with a set of coordinates for the atoms, will be published elsewhere." So because one DNA strand, you can predict what the other strand will look like, because again, it's anti-parallel and A's always must bind to T. They assumed that you can write another strand based on the first strand, and therefore they're saying that they can postulate a possible copying mechanism for the genetic material. So now let's revisit the structure of the double-stranded DNA molecule. And we see, if we take a individual nucleotide on the 5' carbon, we have our overhanging phosphate group. And on the 3' end, we should have that hydroxyl group hanging off at the end, because we don't have another phosphodiester bond from the next nucleotide to fill its place. And on the other strand, it has to run in the reverse direction. So the 5' end is actually at the bottom, and the 3' end of this DNA molecule is at the top. And so this is called anti-parallel, the way they're situated, because they run in the same plane, but in opposite directions. And they are also called complementary because of that feature that we discussed about A's always binding the T's. So if I was to block this half of the DNA strand so that you could only see one strand, you should be able to look at this other strand and be able to draw this strand without ever seeing it. And as a reminder, again, the phosphate group should be on the 5' end, and the sugar should be on the 3' end. So what that means is that one strand tells the story of the other, and that this provides a mechanism for copying one strand into a new strand. If the existing strand of DNA contained the following nucleotide sequence, CAT, CAT, GACT, what would be the sequence of the new strand of DNA synthesized by the cell? Go ahead and take a moment and see if you can figure out what bases you would write for the other strand. Okay, if you took this template strand and you tried to tell the story of the other strand, it should look something like this. You should have a GTAG, TAC, TGA. That's because C's always bind the G's and A's always bind the T's. And you can work all the way down the line here and write the new letters. Okay, so how is DNA packaged in prokaryotes and how is it packaged in eukaryotes? Prokaryotes have a relatively simple way of packaging their DNA. As we've discussed, it exists as one large circular chromosome, depicted here in red, and we can have multiple small circular plasmids, which are extra chromosomal pieces of DNA. And the bacterial DNA is stored in what is known as the nucleoid region. This is not a nucleus, and so the nucleus is not contained, or the nucleoid region is not contained in its own organelle that is membrane limited. In eukaryotes, they're much different. They tend to have multiple, not always, but they tend to have multiple copies of the chromosomes. They are always linear, and they are highly supercoiled around histones. We'll discuss what the supercoiling is in a moment, and they are stored in the nucleus. This is a depiction of eukaryotic DNA structure. So we can start by just looking, at the nitrogenous bases, the nucleotide bases themselves, and remember they're complementary and anti-parallel. However, this is not, actually, none of this is really how it looks in nature naturally. These are sort of cartoonish depictions, but we can kind of extrapolate the ideas from here. The DNA actually exists as a double helix. However, eukaryotes like to package their DNA. So they form what are called supercoils. Supercoils are if you imagine like a headphones, if you spin it too much, or a hose, it will start to wrap around itself again. So instead of just being spun and twisted like this, it starts to twist amongst this back over itself in a single strand. So if we were to zoom in on here, that's what it would look like. It would twist over itself again. And these histones are proteins that help provide stability and denseness to this DNA strand. And so these histones help pack the DNA strand into what is called chromatin. And chromatin can be even more highly compacted into what we call the chromosomes, which many of you have probably remember seeing these chromosomal-like structures. These chromosomes have a certain shape to them based upon the structure, which is unique to each species. So what are the essential functions of DNA? Well, DNA is important for inheritance. So DNA is passed down from parent to offspring. And in order to do so, DNA must be accurately copied into new molecules. This is called DNA replication. DNA is also important in that it serves as a blueprint. So DNA isn't just like a picture on the wall that's nice to have there. It does serve a purpose for the cell. So it stores information about structures and functions of the cell. The information in DNA must be converted into proteins. And to do so, the cell does a process called transcription and translation. For this first lecture, we're going to focus in on inheritance, and then in the next lecture, we'll cover the blueprint. Remember, we can make a copy of another DNA strand by using a template strand. And like Watson and Crick postulated, that's how the cell does it. And so if we took a double-stranded piece of DNA, the cell can make a copy of it. In order to do so, though, it has to gain access to those nitrogenous bases that are in the middle of that double-stranded helix. So it recruits an enzyme called helicase. Recall that enzymes are proteins that reduce the activation energy required to do a molecular reaction. And in this way, the cell is able to orchestrate which reactions happen by controlling the speed at which reactions occur. Anytime you see the ace at the end of a word in biology, it almost always means that word represents a enzyme. So helicase and then ASE, so you should have an indication that this would be an enzyme. This helicase will bind to DNA at a site called the origin of replication. This is where our replication is going to begin. And it separates the strands of DNA. So it will work down and unzip the DNA like a zipper. However, it can't keep the DNA strands from re-annealing to each other. These nitrogenous bases want to form these hydrogen bonds with each other. So single-stranded binding proteins are recruited and bind to these single strands and keep them from being able to reattach to each other. Also, another enzyme called primase creates a short stretch of RNA. We'll discuss RNA later on, but it's another type of genetic material, and this RNA is called a primer, and these primers attach the DNA template strand. So we need primase for both strands, the top strand and the bottom strand. And this primase lays down a primer of RNA that always starts with a phosphate group. And remember, the phosphate groups are attached to the 5' end and ends with a ribose sugar, or in other words, the 3' end. So in that sense, RNA primase will always write 5' to 3'. And it's writing based upon what it's reading. And so it reads in the opposite direction because these two strands are anti-parallel. It must start from the 3' end and move to the 5' end in its reading. So enzymes involved in DNA replication always read the DNA template from 3' to 5' and write the new strand in 5' to 3'. So if you were to look at this depiction of the primases, you should be able to predict which way they'll travel by looking at the strand they're on. If we look at the top strand, it goes 5' on the left side and 3' on the right side. And so that primase should move from right to left because it's reading 3' to 5'. The bottom strand is written, it has 3' on the far left side and 5' on the far right side. And so that means that this primase on the bottom strand is going to go from left to right. It's going to read 3 to 5. And as it's reading, it's going to start writing the new nitrogenous bases 5' to 3'. So it should look something like this. See that 3' to 5' on the top? And it builds the anti-parallel strand 5' to 3', and same with the bottom strand. 5' to 3' is the new strand, reading 3' to 5'. So now that we have our RNA primer, we're ready to continue our synthesis of DNA. Checkpoint 2. What type of bonds are being broken in this step? Okay, now let's start to compare the structure of RNA versus DNA. DNA, as you recall, is short for deoxyribonucleic acid, and RNA is short for ribonucleic acid. So RNA's sugar, the ribose, does not come in the deoxy form, so it has an extra oxygen attached to its sugar. The other thing we should note is that RNA typically comes as a single strand. So we have just drawn here a single strand of RNA. DNA usually comes in the form of a double strand. And finally, RNA has a different set of nucleotide bases. The nucleotide base thymine in DNA is replaced with the nucleotide base uracil. So DNA has CGs and ATs, and RNA has CGs and AUs. Anytime you see a T in a DNA strand, you can always imagine that that would be replaced with a U in an RA strand. And let's look here at the sugars. So deoxyribose versus ribose. As you can see on the second prime carbon, there, instead of having a hydrogen group, there is what's called a hydroxyl group or an OH group. So it's not missing an oxygen in its ribose sugar. Okay, checkpoint 3. What are 3 structural differences between DNA and RNA? So, even though RNA has a different structure than DNA, it is still capable of base pairing between the two. The fundamental difference here is going to be that instead of A binding to a T in RNA, A will be binding to a U. But you can still use the same processes of writing a copy using a template strand. Just know that you're going to replace a T with a U. Some students get a little confused here when they see this A here, or sorry, when they see this T here, they try writing a U for its complement here. But remember that T's don't bind to U's, they are replaced by U's. So T is still going to bind to an A. In RNA, adenine didn't go anywhere. So T would create an A if you read this and wrote a new strand of RNA. But an A would bind to a U. All right, so the primase has created that short stretch of RNA. We called that short stretch of RNA a primer because what it's doing is it's priming the well for, so to speak, of DNA synthesis. We have to observe the overall direction of replication on a DNA strand. So we can see that this helicase is going to zip down this DNA, and the top strand and the bottom strand, as they're unzipped, we're exposing more nucleotide bases. And we have another enzyme we have to keep track of. This enzyme is called DNA polymerase 3. It's called DNA polymerase because it polymerizes new strands of DNA instead of RNA. And it is called 3 because it was the third polymerase discovered. Each one of these polymerases serves a slightly different function. This one is very important in replication. So this DNA polymerase 3 adds DNA nucleotides to the 3' end of the RNA primer. So it's going to be replicating like we'll show in this image. So here DNA polymerase 3 attaches and it's going to replicate in the 5' to 3' direction. So let's go back and revisit this again. So DNA polymerase 3 is capable of polymerizing, but there's a problem in that it cannot do a cold start. It actually needs the presence of these primers in order to begin the process. And so DNA polymerase 3 is looking for these primers in order to bind to the DNA. And just like all other enzymes, it's going to read in the 3' to 5' direction. So if we look at the top strand, here's the 3' end, and to the left is the 5' end. So in order to read, it's got to travel towards the 5' end. So it's traveling down this way, and it's reading these nitrogenous bases as it goes. It's going to create a new nitrogenous base as it reads. So it's reading 3' to 5', and because this new strand is anti-parallel, it's going to create a 5' to 3' strand down here on the bottom. It will look essentially like this bottom strand will look. See how this is 5' and this is 3'? It's going to build a strand that should essentially look like that strand and be in the same anti-parallel direction. So let's look at this again. Here we have that DNA polymerase traveling in 3' to 5' direction, building that new strand in 5' to 3' direction. So there's something interesting going on with this bottom strand. This DNA polymerase 3, it attaches to the primer, but it has to move away from the overall direction of replication because it has to read 3' to 5'. So it's going in the wrong direction, right? So here we have our new DNA strands, which is great, but we're going to have to work on that bottom strand. Something else that is really nice about DNA polymerase 3 is it also proofreads and replaces incorrect bases just added. So if it makes a mistake, it has the capability of catching that mistake and repairing that mistake. All right, so let's continue this DNA synthesis. So imagine our DNA is further unzipped with helicase. As the helix unwinds and the ladder separates, the DNA polymerases can keep creating this top strand continually. So there's nothing really getting in the way of this top strand from synthesizing further and further. However, the process for the bottom strand is not contiguous. As you can see, eventually that DNA polymerase will reach the end here and will drop off. It can't continue copying in the proper direction. So this process will start over when a newly exposed piece of DNA on that bottom strand attaches to a primase, and the primase lays down a new primer. So here's our primase. It's going to read 3' to 5', write 5' to 3'. And now the DNA polymerase 3 can follow in and bind to that primer and read 3' to 5' and build 5' to 3' until eventually it reaches the previous primer, which also causes it to dislocate. And there we go. And this process continues with a complete strand on the top and fragments that are formed on the bottom. And here we go with the DNA polymerase 3. Forming another fragment. All right. So how does biology solve the issue of this fragment that is formed? Well, these fragments are called Okazaki fragments, named after the scientist who discovered them. And what was discovered is that we have one strand which is a little ahead of the other strand. The one strand that can synthesize in the direction of the replication fork is called the leading strand because it's in the lead in terms of synthesis here. The strand that has to synthesize in the opposite direction of the replication fork is a little bit behind and is called the lagging strand. Have no fear though, biology has found a way to deal with this, and it has still another polymerase we need to be aware of called DNA polymerase 1. DNA polymerase 1 fixes this issue by cutting out the RNA primers that were made. So on the leading strand, we have to cut out that first primer, and it does so gladly. And it replaces them with DNA nucleotides, again in the same direction, 5' to 3'. Oop. So let's do our next checkpoint, checkpoint 4. DNA polymerases writes blank and reads blank. Okay, back to the story. We had our DNA polymerase 1, which removed the primers and filled them with, instead of RNA, it replaced them with DNA. However, these Okazaki fragments still have this little nick, this little notch here of missing nucleotides that is the space where the previous DNA polymerase 3 couldn't fully reach the very last nitrogenous base and seal this, this backbone here. So, actually I misspoke. It's not really represented here in this image, but it does fill in everything, but it's missing that final phosphodiester bond. So all of the nitrogenous bases are actually there. It just doesn't have that final phosphodiester bond that links these two fragments together. So we have still another enzyme, and this enzyme ligates those two together, and that's why it's called ligase. So ligase repairs these broken phosphodiester bonds that are referred to as nicks. You can imagine if you came in there with a pair of scissors and you cut one side of the phosphate, you would form a nick in the backbone. So it repairs these nicks in the backbone of the new DNA between the Okazaki fragments. Here's our ligase, and it just seals it up like that. Okay, and now we have a full copy of each the top strand and the bottom strand. So now I'd like to share with you a video that depicts this DNA replication. And this is of a eukaryote, as you can see, with a nucleus. Those are the histones you see, these round balls here, and the DNA, which is supercoiled around them and around each other. Transcription we'll talk about next time is the synthesis of a what's called an RNA strand for turning into a protein. And this is how things are moved in and out of the nucleus through these nucleus gateways. And this is what those gateways look like on the outside of the nucleus. So there is the chromatin. There are the histones or nucleosome complexes being formed. And they can come together in order to form even more dense, an even more dense structure of DNA called chromosomes. We're going to jump forward a little bit. This is mitosis. Okay, here's replication. So here's your DNA polymerase 3. And on one side you can see we have the leading strand moving towards the replication fork. And the top is the lagging strand. So here's the leading strand here just being actively synthesized, and here's the lagging strand building in the opposite 5' to 3' location, running into the last Okazaki fragment and then falling off. Okay, that was a lot of fun. Let's get back to our PowerPoint presentation. In this process that we've just learned about, we have DNA replication occurring on both the top strand and the bottom strand of that double helix, double-stranded DNA. In this way, we preserve the old strands of DNA in the process of making new strands of DNA. And because of that, we call this semi-conservative, and that the new strand of DNA is part old and part new. So we can imagine here we have the two old strands, and in the middle is the synthesis of the new strands in the 5' to 3' direction. This brings us to our last checkpoint, checkpoint 5. Besides DNA synthesis not working, specifically what would go wrong in DNA replication with a broken version of the enzyme helicase, and why? What about ligase, and why? For credit on this checkpoint 5, you need to describe what mechanism will not be operating or functioning properly with those two enzymes, and explain why that would cause issues with DNA replication. So that covers our discussion of inheritance with DNA, namely DNA replication, how DNA molecules are copied and replicated. Now, in the next section, we are going to discuss the blueprint. So DNA is a molecule that stores information. Specifically, it has information there for converting the DNA code into proteins. And this process is called transcription and translation. So we want to know how this information that's stored in DNA is put into action. And why do cells use this system at all? The reason is we want to preserve the master copy. So imagine if I, the instructor, only had one copy of the lab manual. I would never loan that lab manual to any one student to use. Instead, I would make copies of the information for each student and hand them out. DNA is much more stable than RNA because DNA lacks the hydroxyl group, the OH group on the 2' carbon of its sugar base that ribose has. The hydroxyl group is more reactive than hydrogen in the CH bond on the 2' carbon. And therefore, the extra hydroxyl group renders RNA more chemically reactive. If we look at the concept of transcription, it's the synthesis of a complementary RNA molecule from a DNA template. And translation is the synthesis of protein from information encoded in the RNA copy. So if I was to give you a handout on how to do a particular lab activity, you could potentially make, a product in the lab based upon the handout I made from the original manual. This is really important because proteins are the workhorses of the cell. They are largely responsible for encoding traits. And sometimes the process stops after transcription, though, to make a functional RNA molecule. So this adds another layer of control of what reactions take place in the cell. So In order to increase the reaction rate, we use enzymes to do that. Well, in order to make an enzyme, you have to have an RNA code. In order to make an RNA code, you have to be allowed to make it from the DNA code. So this is a whole extra level of regulation of processes inside of the cell. This is called the basic dogma of biology, and that is that DNA is the blueprint in which, through transcription, an RNA script is made. This RNA script then goes to the ribosomes in order to be translated, or in order for that information to be used by the ribosome to make a protein that is based on that code. And we will discuss this a lot more in our second lecture on microbial genetics. And so thank you all so much for joining me, and we'll see you on the next round.